Dear student, today I'm going to teach about the principles of forest disease management. So, before going to study the fundamental principles of forest disease management is avoidance, geographical area, selection of proper field, planting time, and the disease escaping varieties, and avoidance of insect vectors and weed host. And the next one is exclusion, that is quarantine, inspection, certification, seed treatment. And the third one is eradication, crop rotation, and sanitation, roguing, soil treatment, heat and chemical treatment to diseased plant material, use of antagonist, and so on. Okay. And if you see that the other component like protection, chemical treatment, and then immunization, resistant varieties, induced systemic resistance, and the finally one is therapy, chemotherapy or thermotherapy, using this heat energy to kill the pathogen or using the chemicals to manage the pathogen. So these are the outlines. So I will be explaining one by one in detail. The first one is avoidance. How we can avoid the diseases. So in this principle, the first is involvement of practice that prevent the contact between the host and the pathogen. Say that today's example, like if any kind of diseases, viral diseases, to attacking a certain person, the first thing is to prevent the contact. So if you are not going nearby this infected or suffering people, that will avoid the sickness or the diseases. So that we call them as, the first one is prevent the contact. The second one is selection of geographic area. So some pathogens are more prevalent in one certain geographical area, and that pathogen so will not move or attack in another geographical area. Say, for example, some of the temperate zone, and then some of them are, high rainfall zone and in the cold place. So now that pathogen will not move from one zone to another unless like the congenial environment is prevalent. So we must choose which tree species we can grow in a certain geographical area. The selection of proper field, it's very important, like elevated field or low-lying field. If some of the trees, loves water, so we have to plant such a trees in a water-loving trees in a low-lying area. So some of the trees are more sensitive to water, and we have to plant it in a high elevated place. And then planting time, it's very important. Which time we have to plant it. If you plant the trees in an off season, then the pathogen take advantage and it will kill them. And then disease escaping varieties. Some of the pathogen here on the left, you can see the some of the pathogen. So it may start attacking the trees if they have contact. Okay, so we must choose this disease escaping varieties. And if you see that in many cases, the disease, the incidence or the disease severity depends upon the coincidence of susceptible stage of the host and the favorable condition of the pathogen. As you know that for the development of disease, we must know this, the pathogen and the susceptible host and the favorable environment. The pathogen must be a virulent one, and the susceptible host, weaker host. And the third one is a favorable environmental condition. If three things are put together, then only the disease will occur. So this can be see that if the susceptible host, if the pathogen is so susceptible in particular month or a period, so we may change this date of planting or the altering the date of sowing. And then favorable environment condition. If that particular environment condition is not favoring for the pathogen, then we can alter this date of planting with that suitable time. So with this, we can manage the disease. The next one is exclusion. How we can exclude. So the first one is, it means preventing the entrance or the establishment of pathogen in uninfected crops in a particular area. Say, for example, you are bringing some kind of varieties or the tree species from one zone to another. Say, for example, in one state to another. Say, for example, if you are bringing the tree bean varieties from Manipur and bringing to Arunachal Pradesh. So it must be like certified, and whether this planting material have any kind of pathogens. If you are bringing the seedlings along with the soil must be tested, whether it is free from any soil-borne pathogen or not. That has to be tested. If not, if you are bringing any kind of bulbs or any kind of varieties, that has to be checked before planting. So the next one is use of certified seed. While bringing, you have to bring the certified seeds, and the sorting bulbs before planting. Any kind of bulbs, tubers, medicinal plants, that has to be sorted. Like the diseased one has to be discarded. Okay, and sometimes we can check for the presence of virus in the planting material. Right. So any kind of tubers and corms before planting, we must examine them. And in order to prevent the important spread of the plant pathogen into the country, individual states and the state law regulate the conditions under which certain crops may be grown and distributed between states and countries. Say, for example, if I want to bring some kind of forest tree species from other parts of India, like some of the trees, if I want to bring some, neem seedling, want to plant it here. So the state laws, we have to check that whether it is suitable to introduce into the land. If even introduced, will they bring it any disease outbreak? So that we must know. So they enacted the law. These kind of a planting material, you should not bring it. So in that case, we can check the spread of the disease from one state to another state. We can enact some law. So even if the materials are brought, then we must check for its presence or absence of a pathogen in the host. So quarantine. So if you're bringing some seedlings, we must be kept in the quarantine for a certain period. Check whether there's a soil or the leaf would show any kind of symptom like fungal, bacterial, viral disease. So inspection of plants in the field. Okay, so we must, even after planting, we must inspect. If all this, after inspection, if the plants or the varieties are free, then we can recommend for the cultivation. So if it is found to be like infected with the disease, then we must eradicate them on the place itself. That's what that quarantine, it helps. The next one is eradication. In this eradication, if you find that if the pathogen, like a diseased sample, if you see that any bulbs are rotten, any tree species, if you're bringing it is wilted, it is better to eliminate them and eradicate them, right? And that be first one is removal of diseased plants or parts in the rogging to control the virus disease, or cutting the canker tree limbs. Just cut that diseased part and you can apply the Borax paste on the cut end wounds. So in that way, we can remove the pathogen. And then cultivating to keep down the weed host and deep plowing. So after cutting down, so we must burn them if it is there in the weed host, deep plowing, okay, and then bury into the deeper soil, burying into the deeper soil that will kill all the pathogens. So the next one is rotation of susceptible with non-susceptible crops. Like if you are growing some kind of medicinal plants, say some of the species are like Hatunia, if you are planting, if it is more susceptible to Rhizoctonia solanae, and in that particular plot, you don't go for Hatunia cordata cultivation. Then you have to grow some other species, other medicinal species. Thereby, it will eliminate the pathogen. And then disinfection usually by the chemicals, sometime by heat treatment, like spreading of all the waste into the nursery area and then burning them. That it will help to eradicate the pathogen in the field. You must spray on the nursery bed. That will remove all kind of pathogen in the nursery bed or in that area, that we call them as a disinfection, removal of pathogen from that coat. And then protection. The next one is protection. Is use of some protective barrier between the susceptible part of the or the host and the pathogen. Like say, for example, you are cultivating 2 different medicinal plants in an area. So in between, you can grow some barrier crops. Say medicinal plants or herbs are small and 1 feet or maximum 2 feet tall. And you can choose the crop such a way that some kind of cereals you can choose, or some kind of, if it is a tree species, you can grow some Polyalthia, Langia, or in the field level itself, mean in the medicinal plants, then you can grow maize, sorghum. Like so, it is a 5 or 6 feet tall crop, and it can check the infiltration or the movement of the spores. Okay, so that will prevent. Okay. Protective barrier that we call them as. In the most cases, the protective spray or the dust applied in the plant in advance arrival of the fungal spore. Say, for example, if the season, continually, raining, and then after one day raining, then there was a cloudy weather, and some kind of morning dew, and kind of a favorable temperature is there, definitely the Phytophthora is to occur. Like so. We must spray some protective spray, like we can spray some coproxychloride or borax mixture to protect the plants from the pathogen. So in that favorable condition, we can initiate this protective spray. If the congenial environment is there, then we can go for this protective spray. And sometimes it is achieved by killing the insect or inoculating agents. So collect all this, some of the vectors, if it is there, we can collect them and kill them. And sometimes it is achieved by eradication of windbreaks and other mechanical barrier. Okay, so thereby we can check the movement of the pathogen. The principle of protective fungicide, it is to disrupt the natural sequence of infection. So fungicide chemical, fungi means fungi, pseudo means to kill. Any chemical or any substance that kill that fungi, that we call them as fungicide. So that fungicide, if you spray, that will kill the fungi in the sequence of infection. Any kind of, there are a lot of sequence of infection is there. It will kill that, kill and break that particular sequence. These fungicides act on leaf surface to kill the newly germinated spores. So the first one is arrival of pathogen on the host, and then it starts germinating. So some chemical, it will kill the spores. And then some of them are like long chemicals available in the literature. So in the, protective spraying and dusting along with the eradicant chemicals. Okay, so commercially sold chemicals are provided with the instruction note, the compatible possibilities of injury. And so these kind of fungicides, there are different kind of fungicides. It's so specific to the specific pathogen. We must know which kind of fungicides is recommended to kill the which kind of pathogen. So the next one is immunization or the disease resistant. Okay, so even if a person is getting the viral infection, so we'll the other person who has not get any viral infection will get to the vaccination, right? Immunization. In that case, here also, like the kind of immunization, what we call them is cross protection, like inoculating the avirulent strain. That is the strain which is not causing any kind of disease, that we inoculate. It is more applicable for the viral disease. So when you apply this this avirulent strain into the host through grafting, so if you apply through this avirulent strain through grafting, it will enter into the host and it will occupy all the multiplication attacks this trees or the seedlings, then it will not provide sufficient space because there's already avirulent strain is occupied, and which will not cause any damage or the diseases to the trees and the seedlings. Thereby it will protect the disease resistance. See, some of them are natural resistant, which is there on the trees, inbuilt resistance, structural disease resistance, like structural component like spines and hairs and wax coating, and internal like a thick cell wall and so on. Okay, so these we call them as immunization or disease resistance. Okay, so sometimes we call them as disease resistant and tolerant varieties are cheapest, easiest and most efficient way to reduce the disease losses. Like some variety which will not infected by any pathogens, then we can recommend, right? Variety should be selected that possess a resistance and tolerance. So we can choose that varieties based on their inbuilt resistance, like as I said, like thick cell wall, sometimes have more hairy hairs on the leaf, like then wax coating and other chemicals like some phenols and some kind of chemicals which are present in the plant that will inhibit the spore germination and the spread. Okay, and for some diseases such as soil-borne vascular wilt, the virus, the use of resistant varieties is the only means of ensuring the control. And then develop the disease resistant varieties, some kind of like tolerant varieties. Say for example, sandal spike. Okay, and then neem yellowing. Okay, so some varieties, some species, if it is more tolerant, that we can advise to grow. So some species are tolerant to that, and some species are resistant. So we can recommend those varieties. And there's a degree of resistance to the certain disease, some varieties being completely immune and others partially susceptible. Some species, or I can say this, some of the varieties. Okay. And in the variety resistant is the pathogens for the area. It may be released to the growers for immediate production. And then even you can use the modern DNA technology, and it is made easy to engineer the resistant gene and introduce into the species. Thereby we can trace. It is more practice or applicable in medicinal plants. Like these all the gene technology, DNA resistant. We can say that transgenic crops, or we can say that RNA mediated resistance. All these it is more applicable for this annuals and biannuals crops, not in big trees. So intolerance, adverse environmental factors, or nucleic acid sequence that leads to gene silencing of the pathogen. Okay, so it's more studied on these viral pathogens. Use of microorganisms like Pseudomonas butida and chemicals, salicylic acid, to induce a systemic acute resistance. The use of microorganism, as I said, Pseudomonas butida, it is a bacteria which can able to kill some of the other pathogens by producing some kind of toxic chemicals. Okay, and sometimes some chemicals like salicylic acid that induce that is systemic acute resistance, it develops on the plants. Okay, and then activation of plants defense system, like this salicylic acid, once if you spray, that activate to the some kind of production of phytoalexins and kind of phenols, and that helps to control the pathogen, incoming pathogens. And then therapy. The last one is therapy. In this therapy, the It is used to individual plants and cannot be used on the large scale. It is achieved by inoculating or treating the plant with some that will inactivate the pathogens, therapy or the treatment. The first one is a chemotherapy, use of a specific chemical to inactivate the pathogen in a particular infection code or the place. Okay, so that is a chemotherapy. There are certain different kinds of chemicals are there. So that I will teach in other lectures. Then thermotherapy involves the exposure of diseased plants in the hot water or steam or hot air. Okay, so it is more applicable to kill this virus and bacteria. Say, for example, some planting material found to have some virus. So you can immerse them in a 50 degrees centigrade for about 20 minutes. Thereby, all the viruses which are there in the planting material, it will die. Okay, so that will call them as a thermotherapy. Or you can give some time, a steam therapy or like a hot water treatment in the case of a soil. If you're preparing some nursery in the soil, so we can apply this hot water. Thereby, all the pathogen which are there in the soil, it will be killed. Okay, and even sometime it kill all this hot water. It will kill the nematodes and the other pathogens. Okay, which are there in the soil, and sometimes we can these bulbs and culms and tubers that we can immerse it in the water, hot water, 50 degrees centigrade. Thereby, it will kill the pathogens. Okay.